Walking Stick Library presents The Body in the Box with your librarian hostess with the mostest, Eileen Kramer. And don't forget, you can catch this stream live on Twitch. I would like to do the CL1 computer. So I am going to clean up in aisle 9 and we're going to just start it directly without coming inside. I'm going to take this down. We're not going to use it tonight. This is where we were at the beginning. This is a video on YouTube. I'm going to check to make sure I have my...
Just when you thought things couldn't get any worse, something happens that makes you ask the question, what in the world is actually happening? Am I dreaming? Is this all a reality? Well, I'm here to break it to you guys. It is a reality, and I found something that most people won't even believe is real. But it is real, and I'm going to share it with you today. By the way, real quick, before we get started, my X account was deleted recently. And I find it rather suspicious because I had made some videos critical about the Neuralink chip, and we all know who runs the Neuralink chip, who's creating it. Shortly after that, my X account was deleted. It doesn't exist anymore. So if you were following me on X, I'm sorry. However, I did start a new one. The link is in the description. It's at endtimessign. So make sure you go over there, follow me on X, so I can get this account started up again. All right, let's get back to it. Ladies and gentlemen, they've actually done it. The globalists, the technocrat, the mad scientists who are building these things have crossed the line. They built a computer not out of silicone and wires like normal computers, no, but out of human brain cells. That's right, you heard me right. Living human brain cells. This thing is called the CL1, and it's straight out of a dystopian nightmare. And I've got the article right here, and we are going to read it. We're going to tear it apart, and then we're going to connect some dots to Bible prophecy, demonic forces, and what this means for you and your family. So let's get into it. So what is the CL1? Well, I'll tell you one thing. It's not your grandpa's Commodore 64. No. The CL1 was cooked up by some occultists in Australia. Computer scientists in Australia at a place called Cortical Labs. They're literally growing human brain cells in the lab. It's like some sort of freaky Frankenstein experiment. And they're sticking these human brain cells onto a silicone chip. I can't even believe I'm saying this right now, guys, but it's happening right now. They say it's the world's first code-deployable biological computer, and it's going to cost you $35,000. It ships later this year, and get this, it stays alive for 6 months in this box that they have it equipped with life support. They literally have to put life support systems into this computer box to keep it alive. They even got it hooked up to something called the Biological Intelligence Operating System. It's like some sort of fancy way of saying that they're playing God, right? They're creating this living organism inside of a box.
Now, scary things go on in labs. We're CRISPRing things. We're engineering organisms. We're tinkering with DNA. This is right in that strain of stuff, but we're going to take a really good look. So hang on, let this guy finish, and we'll get to what we get to.
I'm not making this up, folks. This is real. Quote, world's first body in a box. Biological computer uses human cells with silicone-based computing. March 6th, 2025. Cortical Labs says the CL1 will be available from June, priced at around $35,000. Australian biotech company Cortical Labs has introduced what it claims to be the world's first code-deployable biological computer, which combines human brain cells with traditional silicone-based computing. The system, known as CL1, was presented at the Mobile World Congress in Barcelona and is being explored for its potential applications in AI and machine learning. The CL1 consists of a silicone chip with lab-grown human neurons cultivated on its surface. These neurons are capable of responding, forming networks that process information similarly to a biological brain. The system is designed to allow a two-way communication, where electrical impulses stimulate the neurons and the responses are recorded and analyzed. To maintain viability of the neurons, the CL1 is equipped with a life support system that regulates temperature, gas exchange, and other necessary conditions. A notable aspect of the CL1 is its ability to learn and adapt to tasks. Previous research has demonstrated that neuron-based systems can be trained to perform basic functions, such as playing simple video games. Cortical Labs' work suggests integrating biological elements into computing could improve efficiency in tasks that traditional AI struggles with, such as pattern recognition and decision-making in unpredictable environments. Cortical Labs says that the first CL1 computers will be available for shipment to customers in June, with each unit priced at approximately $35,000. The use of human neurons in computing raises questions about the future of AI development. Biological computers like the CL1 could provide advantages over conventional AI models, particularly in terms of learning efficiency and energy consumption. The adaptability of the neurons could lead to improvements in robotics, automation, and complex data analysis. However, the scalability of this technology remains uncertain. Producing and maintaining neuron-based systems is significantly more complex than manufacturing traditional processors, and ensuring long-term stability poses additional challenges. Ethical concerns also arise from the use of human-derived brain cells in technology. While the neurons used in the CL1 are lab-grown and lack consciousness, further advancements in the field may require guidelines to address moral and regulatory issues. The prospect of integrating living cells with computational hardware prompts discussions about boundaries of artificial intelligence and human-like cognition.
Beautiful. Now, see if I can get back to, I can't get back to the beginning of this, but we can do a Google search. We want to see the article. And what we're going to do is we're going to make this a lot easier for everybody to deal with. This is going to be what we call a crappy Google search. Hey, how about over here? This is a good spot. Yeah, let's plunk it down. What we're going to do is do a known item search to get that article. Body in a box. Nice little alliterative headline. If English is a very good language for this kind of bounciness. If you're not a native English speaker, just trust me, body in a box. You hear the rhythm? You hear the alliteration, body in a box. It's alliterative, the B and the B. And it's considered, alliteration is a big thing in alliteration is a big thing in English. So is rhythm. Emmanuel, I just need space, okay, honey bunch? And we're looking at the CL1. And we're going to put it together with an and, because we can. And we should find this the article that that our YouTuber at End Times Prophecy was looking at. Because face it, I don't think somebody who's into End Times is going to be a very good computer person unless he's got like a computer show. So we're going to do body in a box. Body in a box. And CL1. And here's the one that he got, first one on top, Tom's Hardware. There it is. What is Tom's Hardware? I have no idea. It's got forums. About us. Whoa, always go in the corners, folks. About Tom's Hardware, our staff, ratings and history. Our mission, Tom's Hardware, is the leading destination for tech enthusiasts of all skill levels. Whether you're building a PC, buying a laptop, or learning how to create robots with your kids, we've got comprehensive editorial resources and a vibrant community. And here we have first names and last names. If I contact you, you've got a looky there, a physical address. Well, folks, we know this is for real. First thing, but I want a better source. Remember, this thing was made by Cortical Labs. Bink. We just have to search for Cortical Labs. We can get the info, we can go directly and get us a primary source. If you were writing about the CL1, the writings of the people who made it are primary. This is, while it's very good, while it's definitely a decent enough source, hello. While this is a decent enough source, while it's a decent enough source, is a secondary source. It's credible. If you're allowed to use web sources, this is probably a pretty good article. But why not go directly, if you're going to use a web source, and get the company? They have a web page too. So we're going to pop this into Google. Bingo! And I'm going to take this down because we don't need any of this anymore. I'm going to check my squawker again. Okay, little squawker. Let's have us a look-see. Here's Cortical Labs, CL1. There it is. That must be the front of it. There it is from the side. This has a clear one. Silicon meets neuron. Real neurons are cultivated inside a nutrient-rich solution, supplying them with everything they need to be healthy. They grow across a silicon chip, which sends and receives electrical impulses into the neural structure. By the way, for those of you who do not know, and look, it moves. Look at this. Yep, you can see the inside of it. It's about the size of a bread box, and it costs $35,000 and only lasts 6 months. I don't think they're going to have very many buyers. Silicon meets neuron, raised in a simulation, connected to the brain, biological neural networks. Okay, I can't get down further. I guess I can. The neuron is self-programming, infinitely flexible, and the result of 4 billion years of evolution, what digital AI models spend tremendous resources trying to emulate. Programmable organic neural networks born on a silicon chip and living inside a digital world. Push the boundaries of synthetic biology and intelligence. Closed loop, high-performance closed-loop system where real neurons interact with software in real time, a robust environment that keeps neurons alive for up to 6 months. Self-contained, all recordings, applications, and life support are on the device, no external compute required. Plug and play, simply plug in cameras, USB devices, actuators, and more to the Cortical Cloud for use in experiments. Programmable bidirectional simulation and read interfaces enable neural communication and networking. Touchscreen enabled, quickly visualize system status, view live data, or run prepackaged assays. Highly extensible, easily connected to the CL1 to external systems via USB ports for broader testing. Animal-free testing. The CL1 was the first biological computer enabling medical and research labs to test how real neurons process information. offering ethically superior alternative to animal testing while delivering more relevant data and insights. Deeper learning, okay. Here we have the research. And we can pick. And they only have 6 articles here. But here we have what brain cells on a microchip can tell us about intelligence. Here's the article. And I notice the date. Ouch. Let me embiggen this for everybody. 2022. But I notice who wrote it. Brett J. Kagan. Keep that name in mind. I'm going to type this over here where we can put somebody. Because we're going to find our person. I've got an author, but this is 2022. Even if it's December 6th, that's a long time ago. Do we have anything newer? Read paper. Oh, ouch. Not fun. Somebody needs to do cleanup in aisle 9 on the website. Here is critical dynamics arise during structured information presentation within embodied in vitro neuronal networks. And this one is 2023. What's the problem here? It's 2025. Have these guys stopped publishing because of trade secrets? And again, we see that wonderful name, Brett J. Kagan. There's a way to find out. Let me get let me get us back. And no, we're not going in this one. We're going to go to Web of Science because they're good for this kind of thing. And remember, first initials and last names, when you put them together, are good. I have an author. His name is Kagan. And when you do this kind of a database, you do it that way. That's just something you know. And this is one of the reasons I'm over here, is this uses one system, not 20 systems. So what we're going to do is just see what he wrote. And looky what I have. Now here we have a patent. Remember we were talking about trade secrets? See the numbers here? This is a patent. It also says Derwent, which is a patent database. Now some of this is librarian lore, but a lot of this isn't. He has a patent. Method for interfacing biological computing device with in vitro biological neurons by neurological computation and experimentation platform. Involves causing electrical, optical, or chemical impulses to be applied at coordinates. Harnessing brain cells in vitro, March 2025. Full text at publisher. Let's go. And here's the abstract. And this is recent. This is the author again who wrote this. So this may be either a primary or a secondary source depending on your subject. For English, writing about this, I would say this gets up in the primary category. Even though science research articles are usually considered secondary, since this is one of the people inventing this, you've got his own words. Harnessing the intelligence from brain cells in vitro requires a multidisciplinary approach integrating wetware, hardware, and software. Wetware comprises the in vitro brain cells themselves, where differentiation from induced pluripotent stem cells offers ethical scalability. Hardware typically involves a life support system. They have to eat, have glucose will work. STEM involves a life support system and set up to record the activity and deliver stimulation to the brain cells. And software is required to control the hardware and process the signals coming and going from the brain cells. This review provides a broad summary of the foundational technologies underpinning these components, along with outlining the importance of technology integration. Of particular importance is that this new technology offers the ability to extend beyond traditional methods that assess primarily the survival and spontaneous activity of neural cultures. Instead, the focus returns to the core function of neural tissue, the neural computational ability to process information and respond accordingly. Therefore, this review also covers work that, despite relatively early state of current technology, has provided novel and meaningful understanding in the field of neuroscience, along with opening exciting avenues for future research. And biological brains exist as the most data and energy efficient information processing systems known. To date, these complex neural systems are the only identified platform to produce generalized intelligence capabilities. Exploring and even harnessing neural systems in vitro would offer a powerful model to understand how complex features such as intelligence arise from their cellular basis and may even be leveraged as a novel information processing platform with advanced capabilities. Yet harnessing intelligence from brain cells in vitro requires a multidisciplinary approach integrating wetware, hardware, and software, previously termed synthetic biological intelligence. Wetware comprises the in vitro brain cells themselves. Early work in this area almost exclusively used primary sources of neural tissue, especially from rodents. More recently, however, generating neural cells via synthetic biology methods from pluripotent stem cells has been positioned as a more controllable, customizable, and scalable approach. Features vital for the successful expansion of technology. Hardware typically involves a life support system and a setup to record the activity and deliver stimulation. Most commonly, hardware involves microelectrode arrays, although optical and chemical approaches have been explored. Finally, software is required to control the hardware and process the signals coming from and going to the brain cells. Ideally, software can function as real-time closed-loop manner, but open-loop reservoir computing approaches have also been adopted. Because of the new challenge of integrating these three essential components, relatively few peer-reviewed studies of developmental systems capable of showing meaningful information processing capabilities of in vitro neural tissues, even in technically simpler open-loop paradigms. And it's going to go on a little bit. Early demonstrations of in vitro neuroplasticity were achieved in cultures of primary neurons taken from embryonic rodents. Some studies did attempt to use ex vivo tissue-mounted approaches, but the poor scalability of these methods appears to have limited the interpretability. Improvements of methods of primary neural culturing did result in meaningful explorations of neuroplasticity and even early learning protocols in primary rodent neurons, mostly cortical cultures, but also hippocampal cultures. Despite some success with these approaches, primary cultures have some key limitations. First, the lifespan of these cultures is best limited to months, typically weeks. Second, it is ethically difficult to justify the use of culture that involves killing animals to generate cells when alternatives exist. Moreover, animal primary cultures do not show the same properties as human networks. While human primary culture techniques exist, this is only linearly scalable. Finally, while some neural subtype specificity is possible with primary cultures, it is technically challenging, difficult to consistently reproduce, especially between research groups, and ultimately limited. Fortunately, the ability to differentiate pluripotent stem cells into neural cells provide an attractive alternative pathway to generate the wetware component. In particular, advances in stem cell engineering have allowed for pluripotent stem cells to be efficiently differentiated into in particular, advances in stem cell engineering have allowed for pluripotent stem cells to be efficiently differentiated into active neurons that display mature functional properties, while other stem cell sources may be used, particularly for non-human cell lines. Pluripotent stem cells from a diverse range of human genotypes will enable early applications of SBI technology, that synthetic biology intelligence technology that focus heavily on improving disease modeling and drug testing. The generation of neurons from human-induced pluripotent stem cell line typically generated from blood or skin of adult donors provides an ethically sound, scalable production for SBI. Okay, so, and it says here, the in vitro generation of neural tissues from human-induced pluripotent stem cells that mimic the stepwise processes of neurogenesis by adding extra extrinsic factors, usually cocktails of small molecule inhibitors, activators, and growth factors, at a specific time point is time-consuming. For example, PSCs can be differentiated into neural stem cells through inhibition of a critical pathway, such as lefty activin, tumor growth, factor B pathways, and bone morphogenic protein signaling, which is commonly termed dual SMAD. The inhibition of the dual SMAD pathway yields PAX6+ neuroepithelial cells capable of forming rosettes, a classic in vitro representation of the neural tube in 2 dimensions. Fibroblast growth factor signaling is crucial for rosette polarization and the expansion and maintenance of neuroepithelial cells, which are neural stem cells with the potential of differentiating into several neuronal lineages. They don't say neurological, they say neuronal. That sounds very British. So what they're doing here, I'm going to come out. What they're doing here is they are getting a tissue sample from a volunteer. That means just a blood draw or a buccal smear, pretty non-invasive. And then they are turning it into a pluripotent stem cell, turning it into a stem cell, and me bad, and they are you'll see what I want to do. That's better. Growing neurons. So we're talking about cells in a dish. We are not talking about taking out anyone's brain. We're not even getting to the point where we have a brain here. It takes a lot more than what they're putting in that little computer to be a brain. Brains have like millions of cells. I don't think they're growing millions of them for each operation. So that's what they're doing, and then they're using them to build a computer. So one more. No one's talking about making making a brain. So that's what they're doing here. And nobody's talking, you're not going to have any of these out in mass production soon because they only last 6 months and are very expensive. So they're not, this is cells in a dish basically. It sounds scary, but again, they're not hurting anybody when they're making this, and they're not creating anything that could possibly be conscious. Maybe later on, but not now. And stuff in labs is scary, but I don't think this particular project at the state it is now is an ethical issue. It's clearly not demonic. If you don't like what goes on in a science lab, stay out of the lab. That's my advice. Thank you so much for watching. And don't forget, you can catch me live on Twitch.